Section 1 is a review of publications that we had this year in 2024. As you know, DSTRI.org is one of the few organizations on the planet that is routinely collecting and publishing on novel dinosaur soft tissue discoveries. And this year we've had 3 publications in 2024. Our first publication was published in Microscopy Today, one of the Microscopy Society of America journals, published by Oxford University Press. Our other 2 papers were published in Microscopy and Microanalysis, which we'll talk about in a minute. But this is a very significant world-first discovery of nematodes in many individuals of dinosaurs that we've studied. We collected 11 different specimens from 9 different organisms, Nanotyrannus, Edmontosaurus, and Triceratops, different individuals in Montana, and all of them had large infestations of nematodes in the bones. Here is an example of a living nematode that was caught by the fruit trap method. You can try this at home. Slice off a piece of apple, put it in a small depression in the soil, water it, and come back in 24 hours. Then you can place the fruit slice in a Bowerman funnel and wash the nematode out and down the funnel into a collection container. Then you can put it on a slide, but you'll need a microscope to examine it. These are the kind of nematodes that we're talking about in these bones in Montana. Now, we routinely put our bones in a preservative at the site, so anything inside the bone gets preserved pretty much right away. This fixative works to our advantage, as one time we collected an entire nematode right out of our decalcification solution. So fixation seems to work best right at the site. And so we have found all of these once living nematodes which were preserved inside these bones when we put them in the fixative formaldehyde, or as we call it, formalin. This is a world first discovery. In fact, others have seen these structures in the bones and have mislabeled them. They've called them intravascular parasites, which are much smaller than the nematodes that we're finding. But the sizes of the nematodes that we're finding in these bones approximate what the size of living nematodes are. And so we published a whole series of these infestations, a world first publication in these dinosaur bones. Our second publication was published and presented actually at the Microscopy Society of America meeting and published in the Microscopy and Microanalysis journal. It was a joint paper between myself and our intern, 16-year-old Jonas A. Cruz from El Paso, Texas. And we published on astonishing soft tissue permanence in surface collected Triceratops shards from Hell Creek, Montana. The point of this paper was that we went out and collected surface shards. We didn't dig for these specimens, they were on the surface. And so we weren't sure what we were going to find, but we found plenty of soft tissues in them, and we presented this actually at the Microscopy Society of America meeting. We also published a second publication in Microscopy and Microanalysis this summer, but this time examining the clots, the blood clots that we find throughout the vascular canals in these bones. And so we noticed that the clots were disrupted, not significantly, but enough to show that they've been on the surface for a while. We're very proud of our publication history, and this is something that we offer our students, as we did Jonas and others who are working with us. You have an opportunity to join DSTRI as a research associate or as an intern and work on these bones that we're collecting in Montana and make world first discoveries and get them published. So this is an opportunity for students to learn these skills, to learn what it takes to do a career in science using microscopy, and to publish at the highest levels even before you graduate high school. So we think that's a really neat opportunity.